Matt is the director of the IIDR, and he is also a Canada Research Chair here at MAC. He is an extremely busy guy, so I'm very grateful to him for accepting the invitation to come and talk to us today about flu and coronaviruses and things like that. So Matt, you know, I was going to say Matt's one of our sort of rising stars at MAC, but that would be incorrect because he's already risen. And is an amazingly busy guy. He, you know, early on in the days of the COVID pandemic, he was invited to be on the National Advisory Committee for Immunization, which is a big sort of national role. I'm sure you were by far the youngest person in that group, and Matt continues to be on that group, which was, you know, crazy in the early days, as he just told me, being just peppered with questions from all kinds of people in high positions all around the country to come up with some sort of policy decisions. So he's still doing that amazingly, and still director of IDR, and has made time to come and talk to us. So Matt's published in many of the top journals: Nature Immunology, Science Immunology, Cell, etc. So Matt, we're very pleased to have you here, and look forward to what you have to say.
Thanks so much for the introduction, Mark. And it really has been a pleasure to have the opportunity to come and speak at Firestone today. This might be, it's definitely at least the second time I've been able to do this since starting at MAC. And obviously, given how near and dear respiratory viruses are to my interests, I think it's really fitting to ensure that there's cross-pollination between the infectious disease groups and the respiratory health group here. I titled my talk today "Forgot About Flu" because in the wake of the COVID pandemic, we actually had for a little while the privilege to forget about flu, and I'm going to talk to you about that. However, as most of you know, flu has now come back with a vengeance. And unfortunately, as someone who's been studying flu in the pandemic space long before COVID-19, what I can tell you is our propensity for pandemic amnesia. And what I mean by that is our desire to forget pandemics soon after they've happened is coming back to bite us already. And we're only days post the WHO announcing that COVID-19 is no longer a global health emergency. Some of you who are younger in the crowd may also appreciate the Dr. Dre reference at the start of the Forgot About Flu title. Some of the other folks may not, though. So the learning objectives today are to review the impact of the COVID-19 pandemic on circulation of seasonal flu, to understand the threat posed by flu in the context of future pandemics, and to highlight the strengths and weaknesses of our current countermeasures. And finally, we'll discuss the need for and the development of new approaches to prevent and treat influenza. So as a virologist, I can't help but start with a token slide of the influenza virion, but this is, it's not trivial in the context of this talk, because the genomic organization of flu is what determines, in large part, its propensity to cause pandemics. Like coronaviruses, influenza virus is an enveloped virus that's decorated with two viral glycoproteins, hemagglutinin, which is shown here in the dark yellow, and neuraminidase, which is shown in the lighter shade of yellow. And the specific subtype of hemagglutinin and neuraminidase that decorate the viral envelope are what give us flu's standard nomenclature. So H1N1 or H3N2, that refers to the subtype of hemagglutinin and neuraminidase on the viral envelope. Typically, hemagglutinin outnumbers neuraminidase on the viral envelope at a ratio of about 3 or 4 to 1. And hemagglutinin is the major focus of our antibody-mediated immunity against flu by virtue of the fact that it serves as the viral entry receptor. So flu binds to sialic acids, which are sugar moieties that are found ubiquitously on all cells. And this is what the virus uses to gain entry. Antibodies that bind to hemagglutinin. So these antibodies are shown here in ribbon diagrams on top of this space-filled crystal structure of a flu hemagglutinin molecule. They can have varying effects depending on the epitope to which they bind. So antibodies that bind near the surface, can you see my cursor or not? Probably not. Okay, that's all right. Antibodies that bind to the top of hemagglutinin, which is where the receptor binding site is located, basically inhibit the virus's ability to bind to host cells and mediate entry. And it's these types of antibodies that our seasonal vaccines attempt to elicit. By contrast, antibodies like CR6261 bind at a site that is distal from the receptor binding site and proximal to the viral envelope. These epitopes tend to be very highly conserved, and antibodies like CR6261 are what we call broadly neutralizing antibodies, which means that they can bind to and neutralize a very large diversity of influenza viruses. However, they work in very different ways than antibodies like HC19 or HC63, which are conventional neutralizing antibodies. And I'll talk about this in much more depth as the talk goes on. So what I'm showing you on the left part of this slide is the diversity of influenza viruses in nature. Humans, over the course of known history, have only ever been widely infected with H1, H2, and H3 hemagglutinin-bearing influenza viruses. We have occasional spillovers of other subtypes like H5s and H7s, but by and large, we only see the tip of the iceberg in terms of flu diversity. There are currently 18 known subtypes of influenza A virus, and 11 known neuraminidase subtypes, which in theory can occur in all combinations and permutations, but which in nature we don't always observe. And that's because there's epistasis, meaning coevolution of hemagglutinin and neuraminidase to ensure optimal pairing. Since hemagglutinin has to bind strong enough to get into host cells, it can only pair with neuraminases which cleave the bond between HA and sialic acid at a rate that allows entry, but also facilitates egress. So if you have a hemagglutinin that binds too strongly, it'll get in fine, but the virus will never release from host cells at the end of replication. So there's this fine balance that has to be achieved between how strong hemagglutinin binds to sialic acid and how well neuraminidase cleaves that interaction to allow egress to happen. In addition to influenza A, which is the type of influenza virus that we often talk most about because it's the one that has propensity to cause pandemics, influenza B viruses also circulate seasonally. Unlike influenza A, though, which has very diverse ecological reservoirs, influenza B is primarily a human virus. So you can see down at the bottom of this phylogenetic tree, Yamagata and Victoria. These are the two lineages of influenza B viruses. And one thing that I don't have time to talk about today that's very interesting that happened in the wake of COVID-19 is the Yamagata lineage of flu B has essentially disappeared. In fact, it may have gone extinct during the COVID-19 pandemic by virtue of the public health measures that were put in place. What some people don't know is that the natural reservoir of influenza viruses is actually aquatic waterfowl, so ducks and geese essentially. But these waterfowl can allow for spillover of flu into many, many other terrestrial species, including things like poultry, songbirds, domestic animals like dogs and cats, horses, swine, and even things like aquatic mammals, so sea lions, as an example. And in fact, we're currently in the midst of one of the largest ecological outbreaks of highly pathogenic avian H5N1 in recorded history. And this virus is spilling over into ecological niches because in birds, flu is a gastrointestinal infection and it's spread by feces. And so where birds and other animals cohabitate, that is a place where there's high risk of cross-species transmission happening. And so what we've seen in places in coastal areas of Western Europe and South America and the Southern US have been huge die-offs of sea lion populations because of course they're in close proximity to coastal birds. In Ontario, over the last month alone, we've seen highly pathogenic avian flu in scavenger animals like skunks and foxes, as well as spillover in domestic animals like dogs and cats very recently, which is very, very worrisome. And I'm going to talk a little bit more about that later. Although many of us probably feel very new to pandemics, humans as a population are no strangers to pandemics. Since 1918, so roughly over the course of the past 100 years, there have been 5 influenza pandemics, the most serious of which was the 1918 H1N1 Spanish flu, and the most recent of which was the 2009 H1N1 swine flu. If you reflect a little bit about the situation in 2009 relative to the situation 10 years later in 2019 when COVID emerged, you will probably not even remember the impact of the 2009 flu on your lives. I think the sort of biggest relic of the 2009 swine flu is in some very old poorly maintained public washrooms. You'll still find hand washing signs from that pandemic, telling you to exercise, thoughtful hand hygiene. In addition, though, to pandemics, we're constantly under barrage by seasonal influenza. And what I'm showing you here is just sort of the typical epidemiology of seasonal flu in Canada pre-COVID. So what you can see is this is the 2018-2019 influenza season. This season was dominated by H1N1. So despite the fact that we have co-circulation of H1N1 and H3N2 influenza every year, as well as flu B, normally only one of those 2 strains dominate. They cause usually upwards of 90% of infections in a given season. The 2018-2019 season was very weird because what you might notice is the dark red lines indicate H1N1 circulation, but the light red or pink lines that are filled starting around week 7 through to about week 19 or 20 are H3N2. So this year we saw a mid-season shift from H1N1 to H3N2. This is very, very atypical. Normally, one of those 2 strains dominates for the whole season. But I think it underscores the lack of predictability of flu. And we really have no idea what determines which of those 2 strains will dominate in a given season. When we think about the burden of flu, it's important to underscore that despite the fact that we've had seasonal flu vaccines for decades, since the 1940s, we still have massive amounts of morbidity and mortality and healthcare system stress as a result of seasonal influenza. In Canada alone, seasonal flu results in an average of about 12,000 hospitalizations and 3,500 deaths each year. And globally, it's estimated that up to 20% of the global population is infected with seasonal flu every year, resulting in 3 to 5 million severe cases, meaning hospitalization, ICU admission, etc., and between about 300,000 and 650,000 deaths. What I want to pause for a minute on, though, is juxtaposing the burden of influenza by age relative to what we've experienced with COVID-19. So at the graph at the bottom, what you can see is the percentage of subtyped flu infections, okay? What I hope you will appreciate is that the vast majority of infections occur in young children between 0 and 4. And there are relatively fewer infections in 65-year-olds and above. However, what I'm going to show you is that the morbidity burden does not match the prevalence of infection. What's also very interesting is that we tend to see much more H3N2 infection in elderly people. And that's problematic because H3N2 infections tend to be more severe than H1N1 infections. And we also tend to see more H1N1 infections in younger people. The reason we see higher attack rates of H3N2 in people above 65 is because when they were young, the first flu that they were ever exposed to were H1N1 influenzas. And that, what we call imprinting, indelibly determines your susceptibility to flu later in life. When we move from looking at number of cases to severe infections, what I want you to appreciate is that there is a really important difference between flu and COVID-19. And that is that although people 65 and above are highly susceptible to both influenza and COVID-19, young children between 0 and 4, who are largely spared from severe acute COVID-19 outcomes, are at very high risk of severe influenza outcomes. So had we experienced a flu pandemic of the intensity with which we currently experience COVID-19, we would have been in big trouble because we thankfully did not see swells of admissions in children's hospitals during COVID-19 as a result of acute SARS-CoV-2. But we would see this in the context of a flu infection because of the different biology of these viruses. What's incredible is the way that our public health response to COVID-19 impacted seasonal flu circulation. So what I'm showing you here on the y-axis are the percentage of positive influenza tests during the 2019-2020 flu season. And what you can see marked in a yellow dot is where stringent public health measures were implemented by all provinces and territories in response to COVID-19 in Canada. And what you can see is that the flu detections dropped like a stone when those public health measures were implemented. This isn't a testing artifact, right? Because this is percentage positive tests. So yes, when public health measures were put in place, you naturally see a decline in flu testing, but this is normalized for the number of tests that occurred, right? And what you can see is that in gray and the hatched lines, we saw a much shorter flu year than what we typically see in the 5-season average leading up to 2019-2020. Now, when we look at the 2020-2021 flu season, the number of positive tests, or the percentage of positive tests is the red line. There was no circulation of seasonal influenza in 2020 and 2021 as a result of the ongoing public health interventions that happened during COVID-19. What I hope this shows us is that what we learned about non-pharmaceutical interventions to mitigate spread of COVID-19 is highly relevant to many other respiratory pathogens that are also transmitted primarily by the airborne route, okay? So this was really an extraordinary observation. Now, though, as we look to the current year, where we've seen the lifting of the vast majority of these public health measures, what we've seen is that flu has returned with a vengeance. We saw an earlier peak of influenza this year than in typical seasons, and that's because sort of its normal habits have been thrown off by the dynamics of the pandemic. So the amplitude of the peak was about the same as what we typically see, but the duration of the season and when the season started was significantly earlier. And this may have been impacted by, in the hatched line, what you can see is this little unusually late peak in influenza that we observed in the season prior, the 2021-2022 influenza season. Our best way to protect against seasonal influenza is, of course, influenza vaccines. And of all vaccines out there, we see the most diversity in seasonal flu vaccine platforms. We have live attenuated vaccines. This is also known to most people more commonly as Flumis. This is the nasal spray vaccine and is really the only mucosal vaccine that has been in widespread use for a long period of time prior to the current pandemic. Recently, we've also seen approval of a recombinant influenza vaccine. In most countries, this is called Flublok, and it is a Sanofi vaccine. In Canada, they call this thing Supemtec, which is a very confusing name and not very descriptive at all. It's the same thing, it's just different marketing. And it's basically purified recombinant hemagglutinin produced in insect cells. Some parts of the world have whole inactivated flu vaccines. None of those are approved in Canada, though. Instead, we have what are called split virus and subunit vaccines. These are vaccines that are grown either in embryonated chicken eggs or cells and are then inactivated by physical and/or chemical means. And we purify basically the viral antigens from those vaccines, okay? Split vaccines are sort of less purified than the subunit vaccine counterparts. What I want to emphasize, though, is that despite the fact that we've known how to make flu vaccines for over 60 years, and there is a massive global infrastructure to produce these things annually, we still cannot make vaccines quickly enough to respond to pandemics, even when all of this infrastructure is in place and we know how to make the vaccine, meaning we know what the correlative protection is. So what I'm showing you here is a slide that I show all of the time to emphasize this point, and it comes from the 2009 pandemic. In 2009, when swine flu emerged, the seasonal flu vaccine had already been produced, okay? But it was recognized that this vaccine was not providing protection from this new pandemic virus, right? By definition. So what happened was pharmaceutical companies and governments around the world scrambled to make a new monovalent vaccine, meaning a vaccine that just contained this new pandemic strain of flu. In blue, what I'm showing you is the number of cases of influenza-like illness. This is, if you looked for flu positive PCR, it would overlay with this almost perfectly during that pandemic. In red, this is the number of monovalent vaccine doses shipped. And in green, the number of doses administered. I hope what you can appreciate from this is that the peak of the pandemic was long over before we even started shipping these doses, much less administering them, right? So for context, because some of you may not be used to looking at these kind of curves, if it were the vaccine, some of you may say, oh, okay, well, like the vaccine's going up, the blue line is going down. Ain't how this works. The sort of plateauing of that blue line is happening independent of the vaccine doses. That green line would need to be shifted over to the exponential part of that blue curve if it were the case that it were the vaccine that were dampening the number of flu cases. This is a slide I've shown now for almost 15 years since I was a postdoc studying pandemic flu in New York. So I told you that since 1918, we've had 4 flu pandemics, the most recent of which was the 2009 H1N1 swine flu. None of those has been as severe as the 1918 Spanish flu, but that has been by sheer luck, not by incredibly advanced medical interventions. The 1918 Spanish flu is widely considered to be the greatest natural disaster in human history. And this is because of the severe impact it had over a very contracted time frame. There have been lots of other horrible infectious diseases that have been scourges on humanity for long periods of historical time, things like plague, smallpox, HIV. But these things cause massive amounts of death over decades, or in some cases, centuries. The 1918 Spanish flu was responsible for the death of 3 to 5% of the global population during that pandemic. If you were to extrapolate the global population in 1918 to today, that would be 370 million deaths over roughly a 2-year period, okay? Again, let's juxtapose that to COVID-19, where we are just about at the 7 million death mark. Now, of course, that is an underestimate of the real mortality burden of COVID-19, but we're not talking orders of magnitude off, okay? Here, right, 370 million is massively different from 7 million. So despite the amazing medical advances that have happened over the course of the last 100 years, I would argue and continue to argue that we are not much better prepared for pandemics, unfortunately. And this risk has really come to a head over the course of the last year. So just as we thought we were getting past COVID-19, there has been an unprecedented global outbreak of highly pathogenic avian H5N1. This has been spread all over the world by migrating ducks and geese, okay? So it probably started as an outbreak in Europe, and as those birds go on their migratory routes, which carries them through North America into South America, those birds carry this virus, and it's now spread all around the world. In Canada alone, we have had 269 outbreaks of highly pathogenic avian influenza virus on poultry farms around the country, resulting in the culling of over 7.5 million birds, okay? The ecological toll of this is severe, and we're only looking in agricultural settings, right? I'm not even talking about the die-off of other domestic, like songbirds, mammals, etc. But the economic implications of this are also enormous, as you can imagine. And if you look to globally, we've lost billions of domestic poultry as a result of these outbreaks. Now, we can handle losing chickens and turkeys, okay? But the real risk posed here is the fact that there are humans who tend these flocks, right? And this puts humans in very close proximity to a very, very serious virus. Unlike seasonal influenza, which causes primarily upper respiratory tract infections and has a case fatality rate well below 1%, H5N1 is very different. And the reason for that is because it contains this mutation in hemagglutinin called a polybasic cleavage site. Normal seasonal influenza requires processing by proteinases that cleave it that are only present in the human lung. But viruses with polybasic cleavage sites can use much more promiscuous and ubiquitous proteases that are present throughout the body. This allows the virus to cause systemic multi-organ infection, including neuroinvasion and outcomes like encephalitis that lead to death. In addition, the type of sialic acid that these avian viruses like are much more dominant in the lower respiratory tract, whereas seasonal viruses, which are well adapted to humans, like sugars that are found in our upper respiratory tract. And that lower respiratory tract infection also leads to much more severe disease because it has a higher propensity to result in serious viral pneumonias and serve as a predisposing event for secondary bacterial infections, which can be very problematic. So we as a sort of medical scientific establishment have been very, very focused in the context of COVID-19 and in its wake on enhancing our ability to respond quickly to new emerging infectious disease threats. In fact, many of you will probably be aware that the 100-day goal is one of sort of the big focal points of the global response to this pandemic. The 100-day goal is essentially a goal to be able to generate new vaccines, drugs, and diagnostics within 100 days of a new infectious disease outbreak occurring. What I want to emphasize is that although this is important, 100 is too pretty a number to mean anything, right? It would never be the like 73-day goal, right? Or the 114-day goal. It's an arbitrary, round, nice sounding number that is within the realm of reality in terms of how quickly we might be able to respond under optimal conditions. For some pandemics, 100 days will do nothing. The pandemic will be blazingly out of control before we ever get to that, and that's the optimal endpoint. What we need to do instead is focus much more on prevention and strategies that will mitigate the effects of infectious disease outbreaks when they happen. We'll never stop them, but we need to mitigate severity to give us the ability to respond. So one thing that we've been, I think, world leaders in at McMaster, thanks largely to folks like you at Firestone, and also to colleagues like Zhou Xing and Fiona Smale at Merck, is in understanding mucosal immunity. And this harkens back, you know, really multiple generations to people like John Bienenstock and Jack Galdi and others who have been really trailblazers globally in this space. So when we're thinking about generating vaccines and the way that vaccines protect us, historically, we've been very focused on antigen-centric vaccine design, meaning we think deeply about what are the targets of our pathogen that we need to go after to mediate protection, but we tend not to think very much or in a very sophisticated way about the way that we administer our vaccines. However, we know and have known for many, many decades that there are important inherent differences in the way that we respond to an antigen that's delivered parenterally, meaning by injection, the way most vaccines are given, versus when we're exposed mucosally. Parenteral immunization, or intramuscular immunization, most often, is typified by the induction of high titers of antibodies, typically in circulation, and these antibodies, by and large, tend to be IgGs, as well as circulating lymphocytes like B and T cells. By contrast, exposure to antigen at the mucosa, whether it be the respiratory mucosa or the gastrointestinal mucosa, is typified by a very different response. It's typified by the production of high titers of local IgA, typically secretory IgA, because this is the type that's enriched at our mucosal surfaces, as well as the induction of tissue-resident T cells, plasma cells, and trained innate immunity, which has become a really sort of vogue and interesting topic over sort of roughly the last 7 years, I would say. Our propensity to vaccinate parenterally is not so much a rational vaccine design consideration as it is a historical vestige of the way that vaccines were first discovered, dating back to Jennerian vaccines. So Jenner is credited, one of the people credited with the birth of vaccinology, right? And it started with Jenner's goal of providing immunity against smallpox using a cowpox virus, right? The way that that immunity was elicited was by taking scab tissue of individuals who, or from animals that had been infected with cowpox, and then scraping that into the skin of naive hosts. This process was called scarification initially, and it elicited an immune response to vaccinia, or the cowpox virus, that was then cross-reacted and protected against smallpox, okay? What many people don't know is that many hundreds of years before Jenner was born, there were civilizations in China and probably India that were vaccinating via a process called intranasal insufflation. Excuse me, I'm dry today. The process is very similar to scarification. They would take scab tissue from individuals at that time who had actual smallpox, grind up those scabs into a powder, and blow that material into the nose of a naive host. That induced, in most cases, a bona fide but relatively less severe smallpox infection. But it was worth the risk given how serious it was to become infected with smallpox were you not already previously immune, okay? So this was sort of how vaccination started, but it wasn't a true vaccine because you had to get sort of infected with the actual virus first in order to gain immunity. It's just that this route of inoculation led to less severe infection because typically the virus had been attenuated and in many cases largely inactivated in those scab tissues. So as I introduced earlier in the talk, the live attenuated influenza vaccine is one of the oldest and most widely used respiratory mucosal vaccines. The way that this vaccine works is that scientists generated what's called a cold adapted strain of influenza virus. So our body's internal temperatures, right, is about 37 degrees Celsius. The way that this virus strain was made was by serially passaging flu at around 30 degrees Celsius, which is around the temperature of our upper respiratory tract. This resulted in a virus that basically can't replicate at 37 and only replicates to a limited extent in our nasopharynx, okay? So that's basically how FluMist works. We've been very interested in understanding the differences in the way that mucosal vaccines protect us relative to parenteral vaccines. And we've been very fortunate to have a long-standing, very productive collaboration with my friend and colleague, Dr. Mark Loeb, who over the course of the last 15 years or so has conducted some really pivotal randomized controlled trials to assess the effectiveness of vaccines in children in Hutterite communities in the Canadian prairies. So the clinical trial I'm describing here is the second trial that Dr. Loeb conducted, and it was designed to compare the relative effectiveness of the flu shot with FluMist in children. So essentially what happened is these Hutterite colonies are colonies of about 200 people. And this type of trial is called a cluster randomized trial. So what happens is an entire colony is randomized for all the children to get either live attenuated vaccine or the inactivated vaccine, okay? And then Mark is able to assess the effectiveness of these vaccines within those colonies. What he found in this trial was that both the live attenuated vaccine and the inactivated vaccine were equally efficacious in children. And that's consistent with our current NACI recommendations around who is able to get these vaccines. So both LAIV and IIV are both equally recommended for young children in Canada. What's very interesting, though, is that we did a follow-up study of the immunogenicity of these vaccines by measuring the amount of neutralizing antibodies that they induce against either H1N1 shown in panel A or H3N2 shown in panel B. And what I hope you can appreciate is that the inactivated virus induces really robust titers of neutralizing antibodies in the serum, whereas the live attenuated vaccine does not, okay? So if you get the flu shot, you get great serum titers of neutralizing antibodies. And if you get LAIV, you really don't get many appreciable neutralizing antibodies in your serum. Nevertheless, both groups are equally protected. So how is the live attenuated vaccine protecting? Well, we were fortunate in this trial to have collected mucosal swabs from these individuals. And what I hope you can appreciate here is that the titers of IgA antibodies found in these mucosal samples correlated super strongly against protection by LAIV, as you can see, with p-values that are extraordinarily low, relative to the inactivated vaccine, where those titers were not protective at all, or didn't correlate with protection at all. That work was done by Jan Ang, an exceptionally talented technician in my lab, who's been with me since I started at Mac and is actually my lab manager. We also wanted to understand, though, how type of vaccine influences the induction of these broadly neutralizing antibodies that I introduced to you earlier. So normally when we get infected with influenza the first time in our lives, our immune system prefers to generate strong antibody responses to the head domain of hemagglutinin, which I'm showing here. When we get reinfected with drifted seasonal variants of flu, we continue to focus on the head domain. So we make a lot of antibodies that are non-neutralizing to conserved epitopes in the head, de novo responses to the new epitopes, but not very many of these antibodies that bind to the stalk domain and have broadly neutralizing activity. However, during 2009, we recognized that people who were infected with that pandemic virus made unusually high titers of these broadly neutralizing antibodies and very modest titers of the conventional neutralizing antibodies. And what we found was that this is a function of immunological memory that has happened in all pandemics in history that we know of. This was sort of the focus of my postdoctoral studies. So in a pandemic, the head domain of flu changes so much that you have essentially no more memory to the head. And left with no memory to the head, the only thing the immune system can remember from prior exposure is the stalk domain. This gives B cells that hold memory to the stalk an ability to outgrow and outcompete those antibodies that normally target the head in a way that we don't see with seasonal vaccines. This is all, though, most relevant in adults. So all those studies that I and some other groups did, around 10 years ago now, were all done in adults. We really don't understand the induction of broadly neutralizing antibodies in children. And so Sergey, along with Daniel, Sergey is a postdoctoral fellow in my lab, and Daniel Celeste, a former super talented fourth-year thesis student, set out to take samples from this clinical trial cohort and ask, how well do flu shots versus flu mist induce these protective broadly neutralizing antibodies in young children? First, what we found is that if we just looked at young children who had been vaccinated sequentially over 3 seasons with flu shot, they made really strong titers of broadly neutralizing antibodies. So that's the red group in panel A. The controls, who were unvaccinated, made very, very little broadly neutralizing antibodies. However, if we look at these two groups and their ability to make antibodies to the head domain of the pandemic viruses, they're equal. So what this tells us is it's not some difference in exposure to the pandemic virus. It really is that seasonal vaccines are capable of inducing broadly neutralizing antibodies sequentially in children in ways that we don't see in adults. We then wanted to compare how well LAIV and IIV do this. So how well does flu mist compare to the flu shot? And thankfully, what we found was that LAIV and IIV induce very similar serum titers of broadly neutralizing antibodies in these young children. And this is good news because it means that one of the unanticipated benefits of vaccinating children seasonally is that we induce this pocket of broadly neutralizing antibodies that's capable of giving them more breadth of protection than what we typically see in adults. And it doesn't really matter which of these two vaccine types that you use. Both are able to do this. Likewise, Both vaccines also induce similar titers of these antibodies at the respiratory mucosa. So we looked at respiratory IgG and IgA, and both vaccines were very similar, despite the fact that when we looked at IgA more broadly in our prior study, we found for strain-specific antibodies, LAIV was much better than IIV. Now, antibody isotype is something that we're also very interested in, because although historically we've really disproportionately focused on the neutralizing capacity of antibodies, we now know that antibodies also work by a lot of other really interesting means, particularly via their interaction with FC receptors on immune cells. So antibodies that don't neutralize can still mediate protection by talking to immune cells that bear receptors that recognize their FC region. A lot of work has been done on this topic by our group and other groups in the context of IgG, but almost nothing had been done to understand how IgA interacts with FC alpha receptors. And the reason for that is largely because mice do not have an FC alpha receptor, and most of these studies mechanistically are done in murine models. So this diagram sort of shows how this happens. So the blue cell here is infected with influenza virus, and during infection, the viral glycoproteins get expressed at the cell surface, which is where the virus assembles. Antibodies can bind to these cell surface expressed glycoproteins and act as a flag for immune cells to come in, recognize that a cell is infected, and eliminate it via mechanisms like antibody-dependent cellular cytotoxicity or cellular phagocytosis. What Hannah did, Hannah was a PhD student in my lab who just graduated last month and is now a postdoc with Shane Crotty's group down at La Jolla. Hannah asked, is there a difference in the way that neutrophils see virus if they're bound by IgG versus IgA? So what she did is she took antibodies from flu-infected people and mixed those together, okay? So she purified out serum-based IgG specifically, or serum-based IgA from the same donors, made immune complexes with virus, and then put those on neutrophils. And then she stained them. And what she saw, very interestingly, was that the IgA immune complexes induced a programmed cell death pathway called mitosis in neutrophils that the IgG immune complexes did not. Sorry, I don't know. Oh, I'm pressing the wrong button. That's the problem. Really interestingly, Hannah went on to show that this phenomenon, not shockingly, is not flu specific. It's IgA immune complex specific. We see the same thing if we look at antibodies derived from people who were vaccinated or infected with SARS-CoV-2, who are HIV positive, or even people who have autoimmune diseases like rheumatoid arthritis and make IgA and IgG specific to autoantigens like citrullinated fibrinogen. Now, while seeing this mitosis was really interesting, the real question is, does it matter? Does it have an antiviral effect? Well, what Hannah showed was that really interestingly, if we stain viral particles shown here in green, viral particles do not like to stick to unstimulated neutrophils. However, when we induce the neutrophils to undergo mitosis by adding PMA, the virus gets stuck in these nets. The nets are composed of decondensed chromatin studded with antimicrobial enzymes. And not only does the virus get stuck in these nets, but if we digest those nets over time and try to collect the virus also becomes inactivated. So those antimicrobial enzymes basically kill the infectivity of the virus over time. In addition to conventional vaccines, another really important tool in our toolkit to protect against infectious diseases are passive vaccines, or what many people just call monoclonal antibodies. These drugs have never really achieved what I would consider to be their optimal effectiveness, largely because they're often administered as therapeutics, and they are just not very good therapeutics. The reason is no direct acting antiviral is a very good therapeutic against flu or COVID because the window for administration is so short. Things that target virus replication in the context of influenza or COVID-19 usually have to be given within 2 days of symptom onset to have any efficacy at all. The same goes for monoclonal antibodies. However, these interventions can be extraordinarily powerful prophylactics, especially in high-risk people who are hard to vaccinate. And individuals who live in congregate care settings, like long-term care, nursing homes, are the ideal use case for these types of interventions. Ali Zhang, an incredible former MD PhD student in my lab, wanted to ask what would happen if we combined broadly neutralizing monoclonal antibody with the most prevalent flu drug out there called oseltamivir. Oseltamivir inhibits neuraminidase. When an immune cell, shown in green, binds to one of these broadly neutralizing antibodies via its Fc receptor, one of the things that we found over the years is that the hemagglutinin makes a reciprocal interaction with sialic acids found on the cell, on that immune cell. So basically, there are two modes of binding. There's the immune cell grabbing onto the antibody bound to the infected cell, but then hemagglutinin on the infected cell is grabbing the immune cell back. Well, I told you at the beginning of the presentation that neuraminidase cleaves hemagglutinin sialic acid interactions, okay? And this destabilizes one of those contact points. So Ali wanted to know if he added oseltamivir to broadly neutralizing antibodies, would we stabilize this interaction and see better therapeutic outcomes? The first thing he did was simply ask, what happens if I add oseltamivir to broadly neutralizing antibodies and immune cells in terms of the immune cell's ability to induce ADCC, or antibody-dependent cellular cytotoxicity? And what you can see in panels B through D is that regardless of what strain and antibody we use, as we add oseltamivir, the curve shifts up and left, meaning that oseltamivir enhances the potency of these antibodies to induce effector functions. We also did this exact same experiment in panel E with an antibody that has a single point mutation such that it cannot interact with the Fc receptor. That mutation is called D265A. It's the same antibody used in panel B, except for it has this point mutation. Now that effect goes away. Okay, so what this tells you is that this relationship between oseltamivir and broadly neutralizing antibodies relies on the antibody's ability to talk to immune cells via the Fc receptor. The big question, of course, was, though, does this matter in an in vivo setting? So Ali set up two experiments. One experiment where he administered either monotherapy alone or the combination of broadly neutralizing antibody and oseltamivir in a prophylactic context, meaning they were given before infection, or in a therapeutic context. And what I should note is that if you administer drug to a flu-infected animal, especially one infected with these highly pathogenic murine viruses that we use, anything beyond within hours of infection, it is near impossible to protect them, okay? The virus is super pathogenic in these mice. What Ali found, though, if you look at panels B and C in the prophylaxis model and D and E for the treatment model, is that the combination shown in purple of oseltamivir and a broadly neutralizing antibody, 6F12, completely protected animals from death and severely attenuated morbidity, whereas either monotherapy alone was significantly less effective. In panel C, you can see we did this experiment with the 6F12 antibody with the mutation D265A that doesn't allow it to talk to Fc receptors. And those animals do much worse when you look at morbidity in B by the black line and mortality in C by the black line, showing again that you have to, it's not just neutralizing activity plus oseltamivir, there really is a synergy that's related to the ability of the antibody to induce ADCC. I think I'm going to skip over this last slide due to time, and just wrap up by saying with a few closing perspectives. So first of all, I hope that I've impressed upon you that prophylactic protection is essential if we want to effectively protect the human population from future pandemics. We will never get there by being faster responders. Flu continues to pose a serious acute public health risk, especially with the unprecedented circulation of H5N1 currently. Both conventional active vaccines, meaning the vaccines we inject or inhale, as well as passive vaccines, meaning monoclonal antibodies, are critical tools to this protection. And although parenteral vaccination is really convenient, it may not be the best way to protect against mucosal pathogens. The rest of these are kind of self-evident. In the interest of time, I really want to thank all the people who are instrumental to this work, especially Dr. Loeb, with whom most of these collaborations were done, as well as Drs. Malarkey, Bowdish, and Kaushik at McMaster, Dr. Cairns at UWO, and my former mentor, Dr. Palese, and my friend and former postdoctoral office mate, Florian Kramer at Mount Sinai, who provided some essential reagents, but most importantly, my amazing lab who did all the work that I had the privilege to present to you today. So thank you very much for your attention. I'll be happy to take any questions.
Thanks very much, Matt. Wonderful, wonderful presentation. So people online, if you have any questions, please pop them in the chat or the Q&A, and I will look out for them. Do we have any questions here from the audience? Dr. Bowdish?
I have two. I'll try to ask them quickly.
Sure.
The some of these base bacterial, I would say more people die of bacterial pneumonia. So I'm curious to know those subsequent influenza pandemics.
Yeah.
The mortality was reduced.
Probably significantly. So definitely the advent of antibiotics and the ability of antibiotics to mitigate the impact of secondary bacterial infections was probably very important for subsequent flu pandemics. There were still aspects of 1918 that were fundamentally different from the subsequent pandemics, especially the propensity of the virus to cause lower respiratory infection. Nevertheless, it is certain that antibiotics have played probably the most important role in reducing morbidity and mortality in people who have infections, including breakthrough infections when there's vaccine failure.
If they do, it's a word of warning for somebody in the antibiotics.
Absolutely.
My second question, okay, is I was looking through some of our partners studies currently have influenza outbreaks. I was surprised at, and I've seen that there's a little uptake of influenza, especially in these off season. Do you have any thoughts on what's happening?
So we really struggle to understand the dynamics of seasonal flu circulation, even at the best of times. Normally, right, the weeks in which we see it are pretty reliable, but there often is offset of flu A and flu B circulation. So, you know, in most seasons, we'll see sort of flu A uptick first and then flu B a little bit later. Not that clear what's going on, but these weird sort of spring waves seem to be a vestige of the influence of the pandemic shifting the dynamic of the circulation period for all these respiratory pathogens. So yeah, not clear what is driving that though.
So we have a question from a participant online. Sudeshna, are you able to ask the question? I think I gave you permission, but then may have taken it away. No, maybe not. No, you should be able. Yeah, okay. Well, I'll read it out anyway. So fantastic talk, Dr. Miller. How feasible or reasonable is it to create multivalent vaccines for multiple influenza strains, e.g. flu A and B? I suppose production based on predictive power would become more complex for multiple viruses.
Yeah, it's a great question. So it's really, of course, there's no limit to our ability to produce multivalent vaccines, but it becomes very, very expensive. So it's really an economic evaluation more than it is like a scientific limitation. We could make a 100-valent vaccine in theory, but the reality is that all of those viruses or all of those antigens have to be produced in isolation and then combined, right? And you need to have a certain amount of each component in order to achieve immunogenicity against that specific component. So I think this dovetails though with something that's really important. And it's a point that I didn't get to make, but I'll take this chance to make it, which is thinking about regulatory frameworks for broadly protective vaccines, which is something most people don't do, okay? And to be fair, I've probably only done this with any sophistication as a result of my experience on NACI over the last several years. So many people ask, it's great to have this ambition to develop broadly protective vaccines, but how will you ever get over the regulatory hurdle of addressing the reality that you cannot test the efficacy of a vaccine against a pathogen that hasn't yet emerged by definition, right? I think the way to address this is that in the context of pathogens that we know have pandemic potential, like coronaviruses and flu, you want a vaccine that you can do a non-inferiority trial with against current vaccines, and then use what we call in vaccinology immunobridging to demonstrate enhanced breadth relative to those vaccines. So that would be a way for us to use vaccine formulations that are very likely to be more protective because they generate what we know to be cross-protective immunity by supplanting our very narrowly protective current sort of seasonal vaccines or variant-specific vaccines in the context of COVID-19. The idea is that these vaccines will not be perfect, okay? No vaccine is going to be sterilizing with one shot perpetually. The point is that you want a vaccine that keeps people alive and out of hospital in the context of a new pandemic to give us that 100-day period to make a new, highly effective strain-specific vaccine. So that's sort of, the framework that I use for addressing this issue in a way that I think is realistic and most likely to be successful in addressing downstream regulatory hurdles that will only happen after phase 1 and 2 clinical trials, right? Where we're only looking at immunogenicity.
Matt, I have a question, which is something that puzzles me, related to memory.
Yes.
Flu and COVID memory. I don't know whether it's a coincidence that COVID vaccines induce very short-term antibody responses in the same way that coronaviruses do. Is that
Right.
just a coincidence? Can you comment on the difference between the longevity of memory in flu versus in COVID, and what's the difference between the two?
Yeah, it's a really interesting question. So I think, and the answer is complex, right? Because it's confounded by the difference between memory duration to homologous virus versus variants or drifted viruses in the context of flu. So the best evidence for the amazing longevity of protection in flu comes from the 2009 pandemic. Normally, I told you that people 65 plus are at very high risk of severe outcomes. During the 2009 H1N1 pandemic, older adults were disproportionately low risk. And the reason for that is because they were exposed to 1918 like H1N1 viruses, which were almost identical antigenically to the virus that caused the 2009 pandemic. So what that tells us is that the protection, at least against severe outcomes, can last for decades, right? In the context of COVID-19, I suspect that the same is true, but it is confounded by the problem of variant evolution, right? So if in flu, we have this phenomenon called pandemic recycling. Basically, population gets exposed to a virus. That population gets older and older, and there's fewer and fewer of them because they die. And now there's a huge portion of the population that becomes susceptible again, which opens the window to a new pandemic. So in addition to worrying about viruses like H5N1, which have never caused a pandemic that we know of in humans, but pose a high risk, people right now are very worried about H2N2. That virus only circulated for 10 years, from 1957 to 68, caused really serious global mortality and morbidity issues. But now, right, anybody who is born after 1968 has never been exposed to H2N2, right? And so this is something that people are increasingly worried about. It's a reason, though, why nobody really expected an H1N1 virus to cause a pandemic in 2009 either. So yeah, this question of durability is really complicated, because I think, you know, sterilizing immunity, I think, is largely a myth that's come from studying immunity to things like flu the wrong way. And by the wrong way, I mean like seasonally over really short periods. Because of course, what happens when you get an infection or a vaccine is you get this big plasma blast response, right, that explodes and then contracts around roughly 3 months. And then you start seeing protection from infection wane, even though protection from severe outcome is really good, because you need huge titers of virus to sop, or sorry, huge titers of antibody to sop up virus to prevent infection. But the memory response, which is slightly delayed, but still will produce new swaths of de novo plasma blasts, can still mitigate severe infection post, you know, an infection actually taking place. There is also no doubt, though, and this is an issue that a lot of groups are interested in right now, that there are antigen and pathogen specific impacts on the relative amount and proportion of memory that gets seeded as memory B cells versus long-lived plasma cells. And right, long-lived plasma cells are what give us that tonic antibody that's in circulation all the time. Memory B cells only produce antibody when re-stimulated, right? If you're reinfected or revaccinated. And we don't understand those dynamics very well at all. But there's clearly pathogen and antigen specific influences there that we just haven't got our heads around yet.
Yeah. Okay, Matt, thanks very much. I think we need to end there. Sudeshna said she's in the middle of a SAC day, which is why she couldn't. Matt, thanks very much. Thanks again for coming in and talking to us. Fascinating work, fascinating insight into these viruses. And a great round. So thanks to everyone online for joining. Thanks to everyone here, and we will resume again next week for research rounds. Thanks again, Matt.
Thank you. Thanks,